We think that parabiosis is 50% mitochondria. In fact, parabiosis was why we started this company. We had a theory. We said, oh, mitochondrial transplant, mitelets in the bloodstream, parabiosis. I put those things together and said, well, maybe the parabiosis effect is really based on all these mitochondria that are getting transplanted. And we've done the tests now with these mice where we have reproduced parabiosis precisely. We are getting mice that show 30% increase in strength, 30% increase in cognition, 30% increase in immune system. And we are just basically doing a type of parabiosis, except it's focused. We just use the mitochondria. Well, the nice thing about the bioreactor approach is that you're doing all the dangerous stuff in the bioreactor. And so I'm not doing that in my own body. I'm doing this in that over there. And I have infinite amounts of energy and infinite amounts of nutrients and lots of time and quality control and technicians. And maybe it's really expensive, but you're not doing it in your own body. You're not risking your life. And you only inject the mitochondria when you're sure they're ready. And you can test them and you can do, so yeah, for all those reasons, an external bioreactor is just a step safer than trying to do it inside your own body. And if you're 90 years old, I don't know how would you fix mitochondria in somebody's 90-year-old body? It doesn't really, I don't think that that's going to be viable.
Hello, Tom. You are the CEO and founder of Mitrix Bio, a company developing mitochondrial therapies to extend lifespan and healthspan. So welcome back to Modern Healthspan, and thank you so much for joining us today.
My pleasure. Nice to be here again.
Thank you, Tom. So, Tom, mitochondria have been rapidly growing as a focus in the longevity area.
Absolutely.
And, researchers are covering their crucial role in aging and healthspan. So could you give us kind of an update on where the industry is overall?
Yeah, happy to do that. And yes, and mitochondria and mitochondrial transplantation in particular, which is our game, it's gaining ground at an extraordinary rate. And I'll tell you more about that. Let me share a deck here, and we'll take a look at some of the details. So, of course, our particular technology is what you would call bioreactor grown mitochondrial transplantation. Okay. And I want to talk a little bit about some of the risks that we see without mitochondria involved in some of these healthspan reversal technologies that are coming down the pike. Before I go any further, I just want to say this is a very experimental. It is not proven. So please take everything I say as a theoretical and unproven. So first of all, mitochondrial research in general for the last 10 years has just been exploding. The amount of new science coming out of mitochondria is extraordinary. And that's why we've got this big boom in mitochondrial treatments, including mitochondrial transplantation, which is the hot topic right now. And what's happening is that what that means is it's really becoming a viable competitor to the main technology that has been out there for systemic health span extension, which what a lot of people are now calling second generation health span tools. And I'll explain a bit more about what second generation versus first generation, but second generation are big genetic changes to your body that dramatically reverse aging as opposed to just, making do with what you have a little longer. I do want to also say that mitochondrial transplantation, there are people doing human trials all over the map right now for various diseases, and we've got human trials coming up for systemic age reversal basically within weeks, potentially. So it is happening now. It is not off in the future. And when I say it's hot, I just mean, there was a big article in The Economist 2 weeks ago. 3 weeks ago, the Jason Calacanis and David Sachs, their big podcast, which is very popular. They had a big section out of the blue. It was very David Friedberg talked about mitochondrial transplantation, so that was exciting. Everybody in the field, we were all buzzing, you know. And then we had our first mitochondrial transplantation academic conference at University of Hofstra University in Long Island, sponsored by Northwell Health, which is the third biggest healthcare plan in New York, where they've been doing lots of mitochondrial transplantation research and finding amazing things. So it is definitely happening. And Mitrix, our company, we've been working on it now for 4.5 years, and we are at the point where we've got research and we've got a volunteer group, we've got celebrities, we've got here's my kind of celebrity list. You know, we've got Tom Randa, who's a very well-known aging researcher at UCLA. We signed up William Shatner, the actor, as an observer to kind of be our celebrity, you know, our celebrity representative. We have a space shuttle astronaut on our board. So it's a pretty big project. Lots of people involved at all over the United States and Canada. So let me just kind of put this in perspective. You know, what I call the Silicon Valley Healthspan field right now, there's a lot of big players putting money into companies. And you've probably covered a lot of these on your show. Most of your viewers will know about these. Altos, Retro, Biosciences, New Limit. Altos got 3 billion, Retro got 1 billion from Sam Altman, who's the guy who invented AI. I mean, these are big players. Jeff Bezos, Brian Armstrong, who's the CEO of Coinbase. So they've got a total, and they're all doing epigenetic reprogramming. They've gotten a total of at least 4.5 billion, maybe more, that's been poured into that. So these are big players. We are the one, you know, they're the elephants in the race, and we're this mouse. You know, we've raised under 4 million from some very wonderful angel investors, and they're all focused on epigenetic reprogramming, which is fixing the methylation on the nuclear DNA. Okay. And again, many people they know about that. That's a hot topic right now. There's also companies on the East Coast that are working on it, you know, in Harvard and other places. And we're coming in with mitochondrial transplantation, which is in a way almost a mirror image. The mitochondria has its own DNA. We're going to talk about this in a couple minutes. I'll take you through exactly what mitochondrial transplantation is. But fundamentally, our bodies have actually two completely different types of DNA. Most people don't know that. We have our chromosomal DNA, which is our own personal DNA in our cells that makes us who we are. That is, you know, created from birth, from the mother's DNA and the father's DNA. But what most people don't know is that we have another whole set of DNA that is not ours. It's a family heritage. We get it from our mothers and our great-grandmothers and our great-great-grandmothers, and those mitochondrial DNA pass down from mother to child. The father's not involved at all, and they may not change for thousands of years. So if your mitochondrial DNA in your body is exactly the same as a mitochondrial DNA in your mother. If we switch them, you wouldn't even notice the difference. So, and you don't read about that in the history books. When we start as an egg, there's almost as much mitochondrial DNA in that egg as there is nuclear DNA. There's about, 3 to 5 billion base pairs of mitochondrial DNA. It's a very small DNA, but there's hundreds of thousands of them that the reproductive system packs into that egg cell. And there's only one nuclear DNA. So it's a very interesting kind of place in the world where we're seeing this variation. And the thing I want to say about this is that we're actually beginning to think, based on our research, and we're not the only ones, that there may be some significant problems with using epigenetic reprogramming and senolytics and telomeres and some of these other techniques that are being used. When you get into people who are over 70 years old, I think there is some risk, because the problem is you can grow new muscles, you can grow new bone, you can grow new brain tissue. That's the goal of epigenetic reprogramming and these other tools. You're making the DNA younger, that makes the cells younger and they grow. The problem is that if you're not at the same time restoring the mitochondria, you're at risk of getting a very serious condition, which we understand in the medical world, where you don't have enough mitochondria to support the load, the energy load that your cells are requiring. And this, I mean, there's plenty of people in the world who have this disease. It's very well understood, and it's not fun. It's tremendously damaging. And so this is why we're really focused on the mitochondrial side, because we think it's going to be mandatory, we think, for doing people over 70, especially. And I'm going to explain why it's so age related. And people say, well, okay, I'm 50, I'm going to get epigenetic reprogramming. That's probably most of the people who are going to try these treatments are going to be over 70. And so we really have to think about them as our customer target base. For example, we do have a class of diseases called primary mitochondrial diseases. These are diseases like Lee syndrome and MELAS and KSS, Kearns-Sayre syndrome. There's a whole bunch of these very rare diseases that people get when they are born with mutated mitochondria. And they're rare, but they happen. And you read about it every once in a while. Some poor baby just dies at age 18 months because their mitochondria are so damaged that the mitochondria can't support the growth of the body, and the body doesn't know what to do. It's expecting a certain energy level and it can't get it because the mitochondria are broken. So it's a very, very risky issue. And this is what we feel like, and you end up, and we know a lot of people who have this. You end up bedridden, you're in pain all the time, you have tendinopathy, your tendons just kind of fall apart, your digestive system fails, you get brain damage, you get strokes, you have seizures, and it is fatal, often fatal. In milder versions, it's not fatal, but you're debilitated for life. So the bottom line is, of course, that is not what we want when we're trying to make people, when we're trying to give them these treatments to make them younger, to make them healthier. Obviously, this is not the goal. So what we're focused on is, what do we need to do with mitochondrial transplantation to hold up our side of this equation? Okay. And by the way, a lot of people talk about drugs and nutrients and exercise in relationship to mitochondria. Yes, those things are important for mitochondria, but when you're getting into your 70s and your 80s, the damage to your mitochondrial DNA is what's mainly the problem. And these things won't do anything about DNA damage. DNA damage is, you can't fix that with drugs. You have to use some sort of regenerative medicine, which is what we're focused on.
For me, feeling my best means being both focused and genuinely calm, pushing hard and operating at full capacity, but without the stress. A non-negotiable part of this is keeping my magnesium at the optimum level. That's why I choose Magnesium Breakthrough from BiOptimizers. It helps me get deeper, better quality sleep and supports my body recovery after training, and I can stay calm and focused throughout the day. Why Magnesium Breakthrough? Most supplements contain only one or two types of magnesium, while Magnesium Breakthrough is the only full-spectrum supplement that contains all 7 forms of magnesium your body needs in one capsule. If you want to try Magnesium Breakthrough, go to bioptimizers.com/modern and enter the promo code MODERN10 to get 10% off any order. They even offer a full 365-day money-back guarantee. So there's zero risk to trying it. And if you subscribe, not only will you get amazing discounts and free gifts, you will make sure your monthly supply is guaranteed. Thank you for your support.
So did that all make sense to you, Richard?
That does. Just is anyone trying both at the moment, even in animals, like epigenetic reprogramming and mitochondrial
We'd
transplantation?
like to. We'd love to do that. Um, in our case, it's just funding. I mean, we've had. I mean, I know people that we could talk to about doing this, and there's a lot of great. There's I mean, the people doing the epigenetic reprogramming research, they're doing amazing research. It's extraordinary, and it's really critical. But again, you can't ignore the two sides of the equation. The mitochondrial DNA and the nuclear DNA, you have to bring them back together. That is our belief. Okay.
Yeah, but it makes sense.
Yeah. So let's talk about how mitochondrial transplantation is a technique that can do this. All right. So let me just give you kind of the basic starting point, and this is for people who may not know this. So, your cells contain typically hundreds of mitochondria. They're tiny little bugs that float around inside the cell, and they actually move around a lot. And they are the power plants of the cell, and that's literally what they do. They have thousands of tiny little molecules on them that are actually spinning turbines, little molecular turbines, and they are generating power. And all the power in your cell comes from your mitochondria. And if they were to stop right now, you would drop dead. I mean, that's just, you would cease to exist instantly. So we are maintained by our mitochondria. And by the way, we are typically about 10% by weight mitochondria. So it's not a small operation. That's the power plant of our bodies, and it is a big, complicated, heavy duty power plant. And it's burning fuel. It is literally combusting oxygen and glucose as fuel, just as if you went down to a coal plant and went and saw a coal turbine. So the the mitochondria is controlled by mitochondrial DNA, which are these tiny little rings of DNA. There's typically 5 of them along the inside of the mitochondrial surface here, sometimes a little less, sometimes a little more. And those little rings are basically ancient. They're a billion years old. And they have been evolving like crazy because, of course, if the mitochondria do a better job, you're going to run faster and you're going to escape the predator. So this is something that's been evolved very, very religiously by nature over the years. And these little DNAs are just like they're like the operating system, tiny little operating system for that mitochondria. Okay. And what we're finding right now is that as these mitochondrial DNA decline over the decades as we age, this is one of the primary causes of aging, and it is absolutely critical in all of these diseases. So we're talking about Alzheimer's, Parkinson's, ALS, AMD, right? Glaucoma appears to be mitochondrially driven, cardiovascular disease, liver disease, kidneys, immune system, and of course, muscles. You lose 1% of your muscle mass every year after the age of 30. And that's probably because the mitochondria are just slowly degenerating and the muscle is shrinking down to accommodate what it has available. So the concept of mitochondrial transplantation has been invented in the last 10 years, not necessarily as an aging treatment. It really was originally invented as a way of curing specific diseases. It was invented by someone who was doing heart surgery and wanted to restore function in the heart. So, and sometimes the mitochondria are transplanted from other parts of your body, just like bringing over a little bit of tissue. And recently, we're starting to learn how to grow them and how to transplant them from family members and so forth. So the question is, why would an old person have degenerated mitochondria? This is actually a really key question. So what we're looking at right here is a scan of mitochondrial DNA. This happens to be from my mother, okay, who was kind enough to give me a sample of her mitochondria. This actually comes from a urine sample, so it's actually kidney mitochondria that come off from the inside of the kidney and go out in the urine. And what we do is we take 500 or 1,000 little copies of the mitochondrial DNA and we unroll them and lay them flat. And so what you're looking at here, these little gray lines, each strand of mitochondrial DNA is represented by one gray line across the screen. These white spaces are just gaps in the machine. You know, we're using this, what's called nanopore technology to analyze these mitochondrial DNA, and the white places are just gaps. That you don't worry about that. But the black and the little blue dots, those are areas where we actually are finding deletions and errors in the code. Okay? So this is a lot like a hard disk on a computer. Over the years, all hard disks acquire bad sectors, and the operating system tags those bad sectors and works around them. That's part of computer technology. It's really almost exactly the same thing in the DNA. These bad sectors accumulate as you age. So here's mine, for example, I'm 62. I'm actually older than 62 now, but I'm going to say 62. Here's my mitochondrial scan. You can see I've got some deletions. And this is my mom's. Just look, and remember, we have exactly the same mitochondria because I got mine from her. Look at the difference. I mean, you can just see it. At age, typically about age 92, 93, it just the damage rate just begins to just accelerate. So if we track this damage, we find, and we're not the only ones doing this. This is a lab that we brought in to develop this algorithm for us. What we found is that it declines at a steady rate as you age. And that's because under stress, when you're worried, when you're walking down the street, whatever, there's a certain level of error that creeps into the mitochondrial DNA as they replicate. And it's just unavoidable. And if you smoke, of course, the error rate goes much faster. And so you die sooner. Smokers, alcoholics, people who have very high stress jobs, their mitochondria get damaged much faster. And this is all supported by research that's been done lately. They did all this research that showed that stress causes stress chemicals in the bloodstream, and the mitochondria are triggered and they replicate faster, and they don't do as good a careful job at the replication, and so more errors creep in. And that's why stress is very dangerous for our lifespan. Also, we feel like below a certain level is when we start seeing all of these diseases crop up, like Alzheimer's and heart disease and so forth, because your body, when it has a lot of energy in the cells, it can kind of make up for a lot of problems. But once it starts running low on energy, all these potential problems, some of these are genetically wired, some of these are damage that you got when you were younger. Some of those problems begin to grow and turn into a more serious issue. Okay. And this is another reason why, for example, smokers are more prone to a lot of these diseases because their mitochondria are getting weaker. By the way, we don't have it on here, but someone just showed me very convincing data the other day that cancer should be on this list. So, so this is our goal with mitochondrial transplantation. What we want to do is get a whole big dollop of mitochondria that are fresh and new and inject them into you and basically supplement you with new mitochondria that your cells would take up. And we're basically restoring your mitochondria bodily to a younger state. And by the way, this is completely contradictory to all the textbooks. And that's why mitochondrial transplantation is such a fun field, because it's bursting all over the place. People are doing it. It's extremely well proven at this point, but it's so new. So basically, the idea with mitochondrial transplantation is it could be as simple as a blood transfusion. You take a bag of mitochondria. Of course, they have to be properly, set up for this, and you just inject it intravenously. And what happens is the mitochondria float around in your bloodstream, and when they run into a cell, If that cell happens to be in a time of need where it is low on its mitochondrial energy supplies, the cells have a pre-programmed mechanism where they reach out and they grab the mitochondria and they pull them in. And you can see this. I mean, it's been shown many, many, many times. So it is at this point, 10 years ago, it was very controversial, and now it is becoming very well accepted. And not only can you transplant them, but in fact, mitochondria are transferring around between your cells 1 billion times a minute. Mitochondria are incredibly mobile. They move between cells. They are transplanted between stem cells and cells. Stem cell comes along in the bloodstream, sees a cell that's damaged. It actually builds a little nanotube, and you can actually see the mitochondria being sent down the nanotube into the recipient cell. And they have videos of this. So it's amazing. The other thing that we've seen is that mitochondria transplant through the bloodstream. And this is the work that we based our company on. There's these things that we call mitelets. Mitelets are come out of platelets. Platelets are these little immune cells that float around in your bloodstream. They typically cause the clotting. There's about 1 trillion of them in your bloodstream at any given time. They're very small. Every platelet has 5 mitochondria in it on average. And when that platelet reaches the end of its 10-day lifespan, or when it gets activated, it squirts its 5 little mitochondria out in what's called a mitelet, which is a little vesicle. It's like a little lifeboat. And that vesicle has a coating on it that it triggers other white blood cells and other platelets and neutrophils and T cells. If they see it bump along, they will grab it and they'll pull it in, because I call it the rechargeable battery pack of the immune system. It's like one of those electric drills where you're running out of power and the drill is not running anymore, and you pull out the battery and you pop a new one in and you can keep drilling, right? That's what the white blood cells are doing with these little mitelets. And it's not just in the bloodstream. We're now finding the brain. Your whole brain is run with mitelets, basically, because neurons don't manufacture their own mitochondria. When the neurons have a burnt out old mitochondria, they put them in a little vesicle and they send them back to the astrocytes. The astrocytes rebuild it, put it in a different vesicle, and send it back to the neuron. So the neurons don't do any of their own mitochondrial manufacturing because they're specialized cells. So there's this whole class of mitochondrial transplantation that's happening in your brain all the time. Also true for cardiac, and they've even found them going between retinas. So pretty amazing stuff. So what this means is mitochondria can get old. Their energy production goes down, as I showed back here with this diagram, right? The energy production in my 93-year-old mother is much lower than energy production in a 32-year-old person, which is much less than when you were born. It steadily declines. And so the way that we view these mitelets is basically almost like a type of youth serum. It's an internal youth serum created by your body that it distributes to other parts of the body or distributes it between parts of the brain to keep cells young by giving them new mitochondria that are younger, healthier mitochondria. And your bone marrow has, of course, enormous numbers of stem cells. They generate these mitochondria. So you could almost view the bone marrow as a kind of almost a factory that is generating mitochondria to supplement the rest of the body. So what does that mean for a bioreactor? You can't get these from donors. There's not enough donors. You can't go around draining mitochondria out of your teenage kids because they wouldn't like it. I've asked and they said no, so I can't get mine. So you need a lot of them. It's just there's nowhere near enough of these in the world. So the only way we're going to do this, we have to grow them in a bioreactor, and that's what this process is. The current concept is to take stem cells out of your blood. We put them in this bioreactor. We grow them in massive quantities. We extract the mitochondria. We put them in a special coating that we developed that kind of makes them into like mitelets, and then we just inject them back into your body. And instead of having, when you're, let's say you're 90 years old, you might have 50 or 60% of your mitochondria might be damaged. We're going to try to flush out the bad ones and put in a whole bunch of new good ones, and that will regenerate your tissues and give you a lot more energy. I will just mention, I'm not going to go into huge detail, but we've done enormous numbers of tests on animals doing this. For example, we did something a couple of years ago where we took mice that were, middle-aged, late middle-aged mice, 13 months old, and we gave them, these mice, a fatal dose of sepsis, which is, a bacterial infection. And the orange line here is the mice that got the sepsis. Within 24 hours, they were dead. And the blue line are the ones that we gave big injections of these mitelets from young mice. So we took a whole bunch of young mice and we got some blood from them, got blood donations from the young mice, got the mitelets out of those from those young mice and gave them to these old mice with disease. And they lived longer and they fought off this sepsis. We also did that with the flu virus. Same thing. They could fight off the disease much better because we reversed the age of their immune system. We figured it's equivalent of about 30 years younger immune system in a human being. And by the way, you could use this with any other treatment. I mean, you could also give them antibiotics. You could also give them antivirals. But the point is, this is giving them an immune system that they don't have otherwise. If you're 85 years old and you get COVID, you're going to end up probably in the ICU because your immune system is so weak. In theory, we could temporarily give an 85-year-old person the same immune system they had when they were 40. So we've also done a whole bunch of work on eyeballs. We've put them into retinas. We've put them into tested it in neurodegeneration, where we've seen these big boosts in the hippocampus energy levels, which indicates that we've reversed Alzheimer's. And by the way, I just want to say, we're not the only ones doing this. There's lots of people doing these kinds of work right now, all over the world. And as I said, there's now 3 human trials happening to help kids who have damaged mitochondria. There have been human trials for strokes. There are human trials being planned for heart attacks and burns and wound healing. And so there's all kinds of things going on right now with mitochondrial transplantation. Okay. So anyway, that's what that is. And we're the one team really, we're the one who's really pushing the aging reversal thing, because again, we're trying to bring this up to the point, mitochondrial transplantation up to the point where it can be partnered with epigenetic reprogramming.
A couple of questions that I was thinking about while we were going through that. So I think, so the way you described the bioreactors and the way the Mitrix system would work, it's using autologous.
That's right.
So has there been any work on whether using other people's mitochondria would have an allergic reaction, or is that something that you're moving towards?
Everybody's working on that. Okay. I can say that right now, no one in the mitochondrial transplantation field, I mean, we talk about this constantly because it's the obvious issue. Nobody has ever seen any kind of allergic reaction of any kind. However, that being said, again, most of them are fairly small application in localized areas. We're the ones really pushing this idea of doing systemic, large scale. So when we do our human trials, which we're ready to do, I mean, I'm ready to push the start button next week to start human trials. It will be very, very, very careful trials. And we have a bunch of volunteers, and we'll start with really small injections and look for reactions and so forth and so on. So But people who get stem cell treatments down in Panama, I mean, they can get allergic reactions. That happens even with your own stem cells. So it is a known risk of any type of regenerative medicine. But I actually think that mitochondrial transplantation, autologous mitochondrial transplantation, is going to be extremely safe because they're your own mitochondria. We're starting with that. Uh, that will probably be a lot more expensive because you have to grow individual people's mitochondria. That's hard. But we're starting with that just because it takes one variable off the list. Probably in 5 years, we'll start looking at doing allogenic, much larger scale bioreactors. Perhaps we can create more common types of mitochondria that you can use on larger parts of a population, for example. Okay. That would be allogenic mitochondrial transplantation.
Okay, that just gave me a thought. Well, there was a couple of thoughts actually.
Yeah.
So we just talked to a person who did stem cells, and she is using third-party stem cells, so
Yes. Yes.
allogenic, but they're very young stem cells.
Yes.
So so the
Which means
stem
they have
cell,
young mitochondria. That's smart because you're getting young mitochondria and young stem cells. Okay.
yes, but the stem cells are from the placenta, I think. So
Yep.
they don't have allogenic markers on them yet, but they are still using those, yeah, like you say, young mitochondria from them, from the mother. And so
The
it would be
placenta is probably the only viable source of these things right now. Is
yeah.
placentas are actually an excellent source of mitochondria. The problem is that person got the wrong type of mitochondria. They got a different. Every mitochondria has a haplogroup, and there's 23 major haplogroups in the world, or 26. I'm R21A, and you're probably H15B, and people in Asia tend to be ABCD. They all came from Africa 200,000 years ago and spread throughout the world with human migration. And we got these subgroups of mitochondrial haplotypes. And the problem is, in small amounts, it doesn't matter so much if you have some different haplogroup. But if you start doing really large quantities, we don't know what's going to happen because the programming between the H haplogroup and an A haplogroup, the programming might be different.
Okay. So
There's a risk to large scale stem cell and exosome therapy that nobody's really thinking about right now.
that is correct. But that is interesting. And I was thinking, we have seen that, young blood plasma, and which is especially like the exosomes from stem cells, because I was talking to another guy, and he said
And
that
platelets.
yeah, the exosomes from stem cells are far more effective than just young exosomes from other people, from other cells. So yeah, I mean, I guess the mitochondria are part of the benefit that's coming from this young blood.
We think that parabiosis is 50% mitochondria. In fact, parabiosis was why we started this company. We had a theory. We said, oh, mitochondrial transplant, mitlets in the bloodstream, parabiosis. I put those things together and said, well, maybe the parabiosis effect is really based on all these mitochondria that are getting transplanted. And we've done the tests now with these mice where we have reproduced parabiosis precisely. We are getting mice that show 30% increase in strength, 30% increase in cognition, 30% increase in immune system. And we are just basically doing a type of parabiosis, except it's focused. We just use the mitochondria.
Do they cross the blood-brain barrier,
Yes.
mitochondria? They do.
Yeah. Many people say no, they don't believe it, but we've seen enough evidence. I believe they do. Yeah. Remember, who was there first, the barrier or the mitochondria? Okay. Those mitochondria are 1.2 billion years old. When the blood-brain barrier was developed 900 million years ago, or whenever it was developed, The mitochondria were the ones in charge of developing it, shall we say. So they have the key. They can go anywhere they want because they were the ones who built the system. It's kind of a most biologists don't like that kind of logic, but that's the way I look at it.
Yeah, that's a maybe you could stop sharing your screen, and then
Mm,
I think if you finished that,
yeah,
yeah, cool, yeah,
yeah. So
yeah.
um,
I mean, kind of
I
along
want to say,
those,
can I
yeah.
just say one last thing about stem cells,
Yes, please.
just to finish up? You were talking about stem cell therapy. If you look at the quantity, of course, stem cells are mitochondrial donors, and so a very, very large part of a stem cell therapy, and they're a type of exosome. Those exosomes, one of those, some of those exosomes are carrying mitochondria. Okay, so if you're getting exosome treatment, by definition, you're getting mitochondrial treatment. Um, but our focus is just concentrating that 1,000x. So one of our treatments, in theory, should be the equivalent of 1,000 stem cell treatments.
Yes, right. Yeah, that would. Yes,
And that's
especially
what
with.
we think is needed to get a 10 or 20-year age health span reversal, that kind of quantity.
As I was studying mitochondria, one of the things they do is they are responsible for deciding when cells apoptose. Well, it's not a verb. When they die,
Right.
it it seems strange that a cell would outsource that to something else.
Well, they spent 200 million years working it out. There's probably a good reason. I mean, that is the way that I think about this, right? Not only that, there is research from last year from Columbia University, some brilliant research that shows they believe now there's some people claiming that the majority of the information processing in the cell is actually happening in the mitochondria, not in the nucleus. The nucleus carries all the data and all the designs, shall we say, but the actual number crunching of the cell is happening in the mitochondrial DNA, where there's a lot of energy because number crunching takes a lot of energy. And so it actually makes sense that it would happen there where the energy supply is right next to it. So the nuclear DNA and the mitochondrial DNA are sending packets of information back and forth. Between the two of them, they are making the decisions about what the cell should do at any given time.
And that very muchly that very much could be true. The mitochondria are amazingly, well, they're
Very
just amazing.
controversial, obviously very controversial. So many people will disagree with that. So
Okay. So you mentioned uh, Mitrix trials. Could you
yeah,
talk a little bit? So you're doing can you review the trial you're doing, and where are you with that?
right. So of course, the biggest issue right now is that the American healthcare system is, and the pharmaceutical and biotech world is kind of in flux, as we know. Funding is being cut left and right. There's almost no funding from VCs. Funding for VCs is just shut down completely. We're small enough that that doesn't affect us. And of course, the big guys with the big war chest, they already have all their funding, so they're okay. Um, so what we are doing is figuring out how to do human trials in a lower cost fashion. And we have a whole set of volunteers, most of whom are 70, 80, and 90 years old. Our oldest volunteer is 90, and he's written 300 papers on quantum physics. He was 50 years as a physics professor at University of Washington, and he's written 3 science fiction books. So this is a guy named John Kramer, who's our 90-year-old volunteer. So we have some really prominent people who are jumping into this and like, hey, I'll give it a try, right? So we're going to start small, as I said, and at the same time, we have to scale up the bioreactor technology to grow larger and larger quantities. But I think it'll be about a year of work to really get through these safety trials. There are a lot of countries all over the world who are interested in this, So we could do some of it in the United States. We might do some of it in Australia or Japan or Panama at some of the stem cell clinics in various places. It's very similar to stem cells. And so we could take advantage of that. It's a lot faster, of course, to go overseas for some of this stuff, especially the very first few trials. We have a lot of very high-end doctors involved in this work, but the problem is that it's very hard to do the first trial in the United States because nobody wants to take that risk because the legal situation is very tough. But yeah, but once we get past the phase 1 trials, then things become much easier because there's a lot of states where you can get right to try regulations. You can go to Montana, you can go to Nevada and Florida where they have right to try rules that allow you to do things that have gone through phase 1 trials. So it actually should happen fairly quickly. We're hoping to get this into, some compassionate use trials for specific diseases pretty quickly.
That would be really good, and I shall definitely be watching for that. So you you were part of the XPRIZE, or you were part of a team that was working towards the XPRIZE.
Right.
So can you talk a little bit about your experience at working in the XPRIZE and
Sure.
what
Well,
happened
the
there?
XPRIZE, the XPRIZE has been very exciting. And of course, Peter Diamandis is a known person in the longevity field already, and he's the one who runs the XPRIZE Foundation. And he's done a great job of putting it all together. And then the Saudi Arabians donated a lot of money. Uh, and they just announced last week, they just announced the finalists or the semifinalists, each of whom got a grant for $250,000. We were part of a team. We were part of a collective through a group called the R42 group who submitted, and we didn't win that particular phase, but there were a lot of people. And the issue with the XPRIZE, we didn't pursue the XPRIZE direction specifically because what they're focused on are much more low cost solutions. They're trying to get a lot of people involved, doing a lot of low cost solutions. And what we're doing is not going to be low cost in the beginning. It just isn't. It's going to be very expensive for the first 5 years because it's really heavy technology. And you remember my slide that I showed where these three big elephant companies in Silicon Valley, they got $4.5 billion. The Silicon, I hate to say this. I live in Silicon Valley, and so it's the approach that the big players here take is when they see a big impossible problem, they can throw billions of dollars at it until they solve it. And that's just the way the game is played. And they do that because they figure, well, yeah, but we'll make billions of dollars eventually if we solve it, right? So that's how they got AI to work. So AI is this big thing now. The way they got AI to work was just by throwing huge amounts of money at it. And so they're doing the same thing with age reversal. And the XPRIZE at $101 million. And that's being split up 40 different ways to 40 different groups. It's just not really enough to feed a project like ours. So, and that is, by the way, that's our limitation. I just want to mention, I talk about where we're ready to do human trials. If the XPRIZE had been larger and I felt like we had a good chance of getting it, that would have been great. It does take money for us to do the human trials. That's what we're working on, is getting the funding.
Yeah, that's yeah. It always seems to be the thing. It is just getting that funding.
Yeah, yeah. And again, mitochondria are always a couple of years behind. And so it's definitely, we're getting a lot of interest, but it's a couple of years behind. Things take time.
So you were also working on, and you mentioned a little bit in your talk, other indications, and particularly AMD and neurodegeneration. Do you have anything you can share on those?
Yeah, absolutely. Well, we've tested a lot of things. We've tested neurodegeneration. We've tested some retinal and eye injections, which would be AMD. Actually, AMD and glaucoma are both reasonable targets. We've tested Of course, a lot of people are doing joint and muscle injections. That's very common. You can already get PRP, platelet-rich plasma, and platelet-rich plasma has mitochondria in it, okay? So we're not the only ones. This is stuff. There's been little bits of this, bits and pieces of mitochondrial transplantation happening for decades. But if we do full body systemic, that requires enormous quantities. So, well, maybe we should just do eyeballs instead. That takes smaller quantities, and that's a perfectly good thing to take on, of course, because if people have AMD, they're losing their vision. Also, hearing loss, it's a big deal for anybody getting up into their 70s and 80s. They'd love to get their hearing loss, slowed down. Parkinson's has been a huge target for us because there's a lot of people. Parkinson's is growing like crazy. It's the Parkinson's rates have gone way up, which hasn't even, I've heard, but it hasn't even made it to, I don't think it's made it to the press yet. ALS, which is deadly. So, yeah, the issue again with all these is finding funding. I mean, I have people ready to do Parkinson's studies right now. It takes funding. So,
Because, I mean, you would think is safety an issue anymore? Because you're using, you're using autologous.
you still have to do the safety trials, whether it's autologous or not. You still have to do safety trials. And again, there's been a lot of human trials, but they've been very small. And so if we're going to do larger scale injections like you would need for Parkinson's, you need some fairly large injections for Parkinson's, and you need to do maybe intranasal infusions, which is something people have looked at. You have to show that it's not going to cause inflammatory conditions, and it's really a whole new world. And the doctors are careful, of course, and they should be. So at the end of the day, you have to. I tell people it's like having the very last mountain, Mount Everest, and nobody's ever climbed it. And you can sit here and you can plan, but at a certain point, somebody's got to try to climb it, okay? And you may not make it to the top that first time, but you got to give it a shot. And that's exactly where we're at. We just have to give it a shot. There's no reason to continue with animals. I mean, these animal experiments are very expensive. It's actually less expensive to do them in human beings.
Yes. And yeah, I mean, it's like rapamycin. If you've shown it works enough times, there's not much point in doing it anymore.
Yeah, just move on. Get into the human beings. I mean, I've got all these volunteers in their 80s and 90s. They're retired and they're like, hey, I got nothing better to do. Let's try it. They're always people. There's some real risk takers in the world. And of course, I also want to mention that there are children. I mentioned earlier in my presentation, there's a group of children who have mitochondrial diseases. And one of the points I always make when I'm talking about this is that a child born with Lee syndrome, for example, which is a mitochondrial mutation disease, and I know families where they have 3 children that are all born with this. It's a horrible thing. And they die. I mean, it's fatal, absolutely fatal, no cure. Their mitochondria are 70 or 80% mutated. It is very possible that the work we're doing in the adults will be transferable to these children. In fact, we're planning on it. And so a lot of these adult volunteers are like, hey, use me as a guinea pig. They think that's great. And a lot of them are contributing money. And they say, if you could, we'll pay for this treatment as adults and then go work on the kids pro bono because those families, they need help. So there's some real social benefits here.
I was thinking, can you use CRISPR or similar technology to modify the DNA of the mitochondria?
Well, that is a big question. There are companies trying to do that. However, there's a little trick there. If you have kids who have a mitochondrial mutation disease, then it's exactly predictable where that mutation is going to be. They know exactly where it's going to be from sector 1501 to sector whatever, right? 1546. And if you do one of these scans, you can see this black, where it's like an entire gene is wiped out in the mitochondrial DNA in 80%. And so it's very easy to say, oh, we could go with CRISPR and just hit that particular spot. And we could clip out the bad part, put in a new part. Or we could just one of the strategies, well, you just any place where you see this mutation, you just snip it and it gets thrown away, and then the good ones eventually are all that's left. And the body slowly rebuilds just the good ones. Okay. Very smart strategy. But with elderly people, that doesn't work because it's random noise. It's randomly distributed. And so there's no way to know where to snip or why to snip. The only way you can do that is you have to, and that's what our technology does. Our bioreactor technology knows how to eliminate the bad ones and keep the good ones. That's part of what our bioreactor does. In fact, that's the main purpose of it. It's not just growing mitochondria. You have to make them younger.
Yes. Or start with a young one and then
Start with young ones.
yeah. Okay.
Which, by the way, we know that happens in nature because that's what babies are. I mean, we wouldn't exist as a species if Mother Nature had not figured out some way of making mitochondria younger, because it would have all run out of steam within 1,000 years. So somewhere in the female reproductive system, there are mechanisms for cleaning out the bad mitochondria and keeping the good ones and making sure that those are the ones that go to the children.
Yes, there must be. Tom, thanks for that update. That was
Yeah.
really interesting. And yeah, I certainly see it's a pity about the cost. And certainly I see why your solution to begin with is going to be expensive.
Yeah.
Because safety wise, a mitochondrial solution seems so much more available. It's so much more, so close, so much closer.
Well, the nice thing about the bioreactor approach is that you're doing all the dangerous stuff in the bioreactor. And so I'm not doing that in my own body. I'm doing this in that over there. And I have infinite amounts of energy and infinite amounts of nutrients and lots of time and quality control and technicians. And maybe it's really expensive, but you're not doing it in your own body. You're not risking your life. And you only inject the mitochondria when you're sure they're ready. And you can test them and you can do, so yeah, for all those reasons, an external bioreactor is just a step safer than trying to do it inside your own body. And if you're 90 years old, I don't know how would you fix mitochondria in somebody's 90-year-old body? It doesn't really, I don't think that that's going to be viable. Because if the body could do that, why isn't it already doing it?
Mhm.
It's kind of, there's a little bit of a circular logic there, I guess.
Yes. Okay, Tom. So if people want to follow your work and or mitochondria more generally, where would they go?
Just go to mitrix.bio, which is our website, and we have a contact form. Send us a note, and we have a lot of stuff on the web. We've got a lot of presentations, and we have a pretty big website with a lot of information on it. So you can find us.
Okay, excellent. So thank you so much for talking to us again today.
My pleasure, and thanks. It's nice to talk to you again.
You too.